When I was at school in third grade, fourth grade, one project that we did in class was to build a microscope to watch the cells in of some tissue, in this case meat. Um, Let me tell you something that I'm not proud of is the fact that I couldn't watch the cell, even that I bought the lenses that they asked me and I tried to organize everything. But after this session, I thought, why it didn't work for me? What can I do or what I should know to make it work? And it was a question that rounds my head for many years until I learned how a basic microscope works and how our knowledge of lenses help us to make a microscope. So I'm Hugo Pim, and in this video I'll show you how the rays of light are organized in a system of two lenses to have homemade microscope. So maybe after this you want to have your lenses and have your own microscope. So let's go. First, what do you think you need to have a microscope? So in order to have a microscope, first thing you need is something very small you want to watch. So in my case, it was a cell. So you can see there that I wrote in red a beautiful cell. It's very small, like meat. I told you that it was meat, so it's a small cell meat. And I'm going to use two lenses. The first question you may ask is, okay, what type of lenses may work for me? So the most common way is to use lenses that are concave. So what is a concave lens? A concave lens is a lens that is formed by two piece of glasses, could be, that comes from a sphere, okay? And you select the two pieces of glasses in this form. What for? Because what you want is that the ray that enters to the mirror gets refracted through a point, okay? So this is what a convex ray, a concave lens work. Concave, not convex, concave. Concave. You know, I'm left-handed and usually mix it, but this is concave lens. So this is how the concave lens works. And you need just to know two basic rules to draw the rays of light in a concave lens. The first rule is that any ray that is parallel to our principal line, in this case this is our principal line, I'm going to put a P. Any ray that is parallel to the principal line, it will be refracted towards the focus. Okay? And the other rule I'm going to use is that any ray that goes to the principal point is here, the center of the lens, is not going to be refracted, so it's going to pass in the same direction. These are the rules I'm going to use to see what happened to my cell after it passes through these two lenses. So let's do it for the first lens that is here. So the ray that is parallel to my principal line, I'm going to refract it towards my lens. Okay, so this is the first thing I'm going to do. And now the second thing is the line that goes to the principal line is not going to be refracted, so I'm going to do this line. Okay, so now you can see that there's an intersection point. So that will be our position and the place of our image. Here I erase the position of the other focus, 3 and 6. 3 and 6. So let me write the focus. I forgot to write the focus here. Okay, so my new image, the image of my cell, is going to be in this position and with this size. Okay. Look at that, I magnify the cell. You see it? I have magnified the cell. So you can see that my new image is magnified. So this is 2 squares, and now I magnify my cell like 1, 2, 3, about 4 squares, okay? 3.5 squares. So I magnify my cell. But now we have another lens. Look at this lens here. So this lens is going to magnify the image we have here. So I'm going to apply the same rules, but I'm going to use orange, you know. So the ray that is parallel, let me use the ruler. Use a ruler when you do this type of shapes. The line that is parallel to the principal ray is going to refract it through the focus. And the line that goes to the principal point is going to pass. Look at that, these two lines are not going to intersect here. Okay? They're not going to intersect in this region. They're going to move and spread. So you need to do lines that are here. So we're going to do imaginary lines. My principal line, my focus line. Okay, I need to move my scale. So again, my principal line. And my focus line. Oh my god. This work. Okay, look at. I changed. Okay, look at the intersection here. So what I did my parallel line to the focus, my principal line, and I got that. Okay, I gotta move everything here, but there's no problem. This will be imaginary lines. These imaginary lines are going to connect on this point, right? So my cell is going to change. So let's do our new cell. You know, our new cell is going to be kind of this. Beautiful. Okay. Oh my god. You see? It's changing our cell a lot. Fantastic. Okay, so look at, due to two lenses, what we have changed is this small image of 2 squares by this image. So this image is about 10, 3, 15, 16 squares, about 16 squares. So what we did is to change a 2 square image to a 16 square image. We magnify this image about 8 times. So imagine you have a cell, a very small cell, and you use two lenses, two concave lenses that you can buy in any you can buy lenses any place, you know. So using two lenses you can magnify it 16 times yourself, so you may see things bigger. And we're just using a small principle, concave lenses, and we're using the fact that the ray that goes parallel to the principal line refracted to the focus, and the ray that goes to the principal line is not refracted. These are the only two things that we need to know to have our microscope. Now, of course, how to do it, how to make it better. So you need to know how to measure the focal length, this length, this length, this distance. Okay, you need to know how to measure this distance and how to move these lenses in order to have this type of images. But so hopefully this is not part of this video. I hope that you understand how a basic microscope works, and I hope that this video helped you to see how science is applied and you can apply it to make new devices. So just remember, if you have any comment, just write it. If you like this video, please like it or subscribe to my tube. And also remember that all my social media are using the same name. You can see it here. And if you enjoyed this video and you want to see the numbers behind of how to calculate them, just write and I will do a new video explaining that. I also encourage you to check other videos about lenses that I'm going to post right down here. Okay, so see you, bye bye, have a nice day.